In Chapter 18, we are now talking about the adaptive immune response. This is dealing with response to very specific foreign entity. It could be a bacteria, it could be pollen, but it's the way the body responds to very specific foreign thing. Previously, we talked about the innate immunity, which was non-specific. We also mentioned for the innate in the previous chapter that involves the first and second line of defense. The adaptive immunity response is the third line of defense. Response is only going to occur after you've had that first initial exposure. So you have to have that first initial exposure to the pathogen. It's very specific. There is a memory component of this, which is different than the other two, so that if you ever see that specific thing again, you can have an immediate response, oftentimes where you don't even know that the pathogen was in you. To give you an idea as to how specific this response is, pollen from, say, a white pine is different than pollen from a ponderosa pine. Your body can tell the difference between the two. The influenza virus, the flu virus, has different markers on its surface. We use those protein spikes to help identify it, and we give it a letter designation. We've mentioned this before. There's an H and an N. So oftentimes for flu, you'll see it designated as, say, H1N1. This adaptive response is so specific, it can tell the difference between H1N1 from H1N2. So when I say it's specific, I mean it is very, very specific. There's two components to it. There's what we call the humoral immunity. That involves the B cells. And then the other component is the cellular immunity. That involves the T cells. So we're going to discuss all of that in this chapter. Something else we will talk about are vaccines at the very end and how that plays a role with this adaptive immune response. Benefit of vaccine is that once you've been exposed to a potential pathogen, if you ever see that pathogen again, you're going to respond to it. The value of vaccines is that what we're finding over the years is that you don't have to expose an individual to the entire pathogen. You just need to expose it to just a part of the surface of it. This is a picture showing before the polio vaccine became available. Polio was devastating. It usually occurred in the summertime because it's caused by a virus. It's transmitted oftentimes in water, and so it was very common for people who would be out swimming in a lake and ponds, in a bay or whatever, that before the vaccine, there's always kind of a fear amongst parents of like, oh, you can go swimming, but please be careful, because the number of cases of polio increased in the summer, and it was often associated with contaminated water. You never knew ahead of time who had a serious response to the polio virus. Some people had a very mild response, and there was no lasting repercussions from it. Some people had kind of intermediate. Maybe they had a limp. Maybe they had a hand they couldn't fully use because it was partially paralyzed. Some people with extreme results would have paralysis where basically they could not breathe on their own. And so the only way they could survive at the time, what they made were these, as you can see, these metal containers that were referred to as the iron lung. And as you can see, the only thing sticking out is the individual's head right here. That's the way these people survived. That's the only way they could survive. And so the advent of vaccines has been tremendous in help reducing the number of cases of polio, so nobody has to live like this anymore. If I remember correctly, just like within the past year, the last individual in an iron lung passed away. I had a professor who, when he was in the military and he worked at Walter Reed Hospital, he talked about working on a floor where he was responsible for 16 patients in an iron lung. And I mean, it made an impact on him, obviously, you know, to see the quality of life that they had. So we've come a long way. When we are talking about whether you're talking about the effect of vaccines as it relates to infections, the immune response, we often talk about a first response and a secondary response. So the primary response, that's a first response that you have the first time you ever have exposure, first time you ever come in contact with that antigen. The antigen is the foreign substance. The secondary response, this is going to be any subsequent. So you have to have that first primary response first. It's going to activate everything. There are memory cells that are made, as you're going to see, so that when you have any subsequent exposure, those memory cells are activated and you're going to have a very fast and much stronger response. Like I said earlier, you may not even know that you have an infection. And that is shown here on this particular graph, that it takes a little bit of time from the initial exposure to get everything up and running. So there's a little bit of a delay here, but you finally get your response here. And then after you've managed to clear out the infection, the number of antibodies, the response goes down. Then you have a secondary response. This may be weeks, this could be months, it could be years later, but look how quickly you have this response. There's no lag time like you had over here. And look at the height of the response as compared to the first one. It is much, much stronger. That's all because of these memory cells. You already have, it's kind of like thinking of it this way. You already have all the legwork done. You have to work on a project. Well, you all have all the basic work done. You just need to put it together and boom, you go. So antigens, like just those are the foreign substances. Antigens will activate your adaptive immune response. Some people refer to them as immunogens. They will trigger the production of antibodies. So some people get confused between antigens and antibodies. Antigens are foreign. Antibodies are what you make in response to that foreign antigen. Now, that foreign antigen, that foreign substance, you're not usually responding and making antibodies against the entire, say, organism. Let's say you're talking about a bacteria. You're only making antibodies against a portion of it. There's something on the surface that's going to trigger the antibody production. Usually those are some type of protein. Proteins make very good antigens. So you know how we are in microbiology. We're going to give it a name. Epitopes, this is just referring to the substance that's on the surface, that particular substance that stimulates the antibody production. Haptenes, this is something that is usually very small. It's a free epitope. It's too small to trigger an antibody response, so you have to combine it with something else. It's kind of like, you're a little too small here, we can't see you. So you need to attach it to something else. So this is an example of an antigen, a foreign substance. And as I said, when you produce an antibody against it, you are not producing an antibody against this entire substance. No, you are just making an antibody against a portion of it. So say just against this pyramid structure. This would be one epitope. This yellow circle is a second different epitope. And then the green column here, this rectangle, this is a third epitope. You, as an antibody, is only going to be made against one of these, not all three, just one. So in this case, if this were, let's say, a bacteria, it is possible to make 3 different antibodies against it. One against the pyramid, one against the circle, and one against the green column. And that's what this is showing. This actually is a benefit to you because you're making 3 different antibodies. You're going to increase the chances of seeing it and having a response and destroying it. So the epitope is just the thing on the surface that's triggering the immune response. It's on the foreign substance. So antibodies, just saying, right now we're kind of describing all the players, and then I'll tell you how they all work together. Antibodies are also referred to as immunoglobulins. They are glycoproteins. That means it's a protein with a sugar on it. When you look at it, it is often described as looking like the letter Y. You have 4 protein chains. You have 2 long chains. They're referred to as heavy chains in the middle. So they go from the very tips of the Y all the way down to the base. They're long and they're referred to as the heavy chains. Think of them as they're long, they weigh more, so they're heavy. On the outer portion of the Y, just along the tips, and there's a picture, so it'll be easier to see in the next slide, there's 2 shorter chains. The shorter chains are referred to as light chains. Think of them as they're shorter, they're smaller, they weigh less. So they're the light chains. Near the very tips of the Y is what we call the variable region. Near the base of the Y is a constant region, meaning it's the same for all of the antibodies. On the very end of the tips of the Y, that is where we have what we call the antigen binding site. That is very specific for each antigen. That's where the foreign antigen will bind. And there's 5 different classes or types of immunoglobulins. So when you look at a picture of it, one of the antibodies or the immunoglobulins, over on the right is trying to show you how it wraps around. It's a protein. You can see the 3D structure of it. Personally, for me, at least the way I visualize things, is better on the left here. The purple chains on the inside, these are those heavy chains I was talking about. So both of these are your 2 heavy chains. These smaller green ones over here on the outside, on the ends of the Y, those are your light chains. The lighter portion, and let me see if this might show up better with a different color. Let's see here. You can tell the difference in the color. So this upper portion here where it's the lighter green and the lighter purple, that's the variable region. That's going to be different between one antibody and another. And then the darker portion, the darker green, the darker purple, that's the constant region. So that's going to be the same regardless of which antibody you're talking about. And then on the very tips of the Y, this region right in here, so you can see a little bit better, this area right here. This is known as the antigen binding site. So when an antibody comes in contact with a specific antigen, then this is where the antigen binds on this end, on the tips of the Y. So that's the structure of your antibodies, or those, like I said, they're known as immunoglobulins. As I mentioned previously, there's 5 classes of these. They're named IgM, IgG, IgA, IgD, and then IgE. The Ig stands for immunoglobulin. IgM is a pentamer, so it has 5 of those Y structures connected together. When you are first exposed to an antigen, that foreign substance once again, first time you're exposed to it, you're going to have production of a lot of IgMs. That's the first one to respond. That's the first one to be made in response to that antigen. Numbers of IgM after a while are going to start to decrease. As it's decreasing, then you see an increase in your IgAs. The IgA is going to last much longer. This has the memory cells. It's going to last throughout until you're done with the infection and even beyond that. IgA is a dimer, meaning it has 2 of those Y-looking structures connected together. IgAs are common in mucus secretions. IgD, we don't really know fully what the function is. There's some guesses, but we don't know for sure. We don't have all the answers. Years ago, I can remember hearing a phrase, the more you know, the more you know you don't know. It's kind of like for every question you ask, there's 20 more questions. Do we know all the answers in any field of science? No, we don't. We're constantly, trying to find answers. And as we answer one thing, then we come up with 20 more questions. So, realistically speaking with you, we don't have all the answers. We don't know yet. IGD is one of those examples. Maybe one day you guys can go into research and you can figure it out for us. Then let me know. IgE. This is involved with parasitic infections and allergies. So this table, which is from your book, shows the 5 different types, what they look like structurally. Do you have to know all the details about them? No. But it gives you an idea of what they look like. Be familiar with, if I were to ask you, what are the 5 types, can you tell me? So great, you make the antibodies. Now what do they do? What's the purpose of them? They're going to bind with the antigen. Okay, so if they bind with the antigen, now what? They can do several different things. They're not necessarily going to do all of these, and they won't do all of these, but they will do usually one of these. Neutralization, opsonization, agglutination, complement activation, or antibody-dependent cell-mediated cytotoxicity. Don't you love us with these long phrases, which we abbreviate as the ADCC. Neutralization. What happens here is the antibody is going to bind to that epitope, that region on the antigen, and essentially it's going to prevent it from binding to the host cell. If it can't bind to the host cell, you're not going to end up with infection. And so, excuse me, this is showing here on the far left, you have this virus right here that has these spikes on it. The antibody is binding, remember on the tips, that's where it will bind to the antigen. So that antibody tip right here is binding to this antigen, the epitope in this case is the end of that spike. And so it's binding to it. If you have all of these antibodies that are bound to this virus, one thing that will happen is that the virus can no longer attach to the host cell. So you just prevented it from infecting another cell. Sometimes it will bind to a toxin. Some organisms, such as the diphtheria bacteria, make a toxin. Well, the antibody is against the toxin. And it basically neutralizes that toxin, prevents it from binding to the cells where it would cause damage. And this is showing it's actually binding to the flagella over here on the far right with this bacteria, once again interfering with it being able to move around and attach to the host cell. So neutralization, it prevents it from binding and attaching to its host cell. Opsonization is when it's going to coat the pathogen with molecules, and if it coats it, that is going to assist, kind of like putting a neon sign on to phagocytes to come here and bind to it and eat it. And so here you have this dark structure right here is the pathogen. Notice you have 4 different antibodies that have attached to it. And that essentially is telling this large structure is a phagocyte. It's a macrophage, which carries out phagocytosis. By attaching the antibodies to the surface of this pathogen through opsonization, what it's doing is, like I say, it's like putting a flag or a neon light on it or something, saying, here it is, here's something foreign, come get it, here's dinner. This isn't part of us, come take care of it. So it's increasing the chances for phagocytosis to occur. Agglutination is when you have cross-linking occurring. Antibodies will basically attach to several of the pathogens. You end up getting these clumps. The larger something is, the easier it is for the body to see it. And in this case, it would be easier for cells in the liver and the spleen to be able to filter out these clumps and say, this doesn't belong here. Hey, phagocytes, can you go take care of this? So in this case, you have the red structures are the bacteria, You can see that you have, in this case, because it is a pentamer, these 5 basically Y's stuck together, that is an IgM. It is attaching. As you can see, they can attach to more than one bacteria at one time. And so you start to get this clumping pattern. That is also going to attract cells that carry out phagocytosis to come attach to it and start destroying it. And like I say, it will also help. The liver and the spleen are involved with helping to filter things out. Complement activation. Antibodies will help to activate the complement cascade. We've talked about that previously in the last chapter under innate immunity, part of the second line of defense. Remember, this is where you have a series of proteins that have to be activated in a particular order. The bottom line is it's going to kill the foreign cell. And so what happens here, how does it get activated? It's the antibodies. Antibodies will activate it. It's going to trigger the inflammatory response, so you're going to increase the blood flow. You're going to be attracting the macrophages to carry out phagocytosis there. Those macrophages that are coming to carry out phagocytosis, you've got some antibodies binding to the cell, so opsonization is working there. You're also going to be triggering the complement cascade, which essentially is going to destroy the cells. The ADCC, antibody-dependent cell-mediated cytotoxicity. This is used especially for parasites that are really big. They're too big for one of your phagocytes to engulf it. Basically, I don't care how big of a mouthful you take, it's just way too big. And so what happens here is several antibodies will bind to the pathogen. Oftentimes this occurs with your larger parasites like worms. So you get several antibodies binding to it. This tends to attract then your natural killer cells. The natural killer cells arrive on scene. They're going to release a particular type of cytokine that this chemical then will go into the pathogen and kill it. And so this is showing, how does a natural killer cell know that this particular cell right here is a pathogen? Because it's attracted the signals from the antibodies. They are attracting, so the natural killer cell comes in, it then releases these chemicals that then will move across and they enter into the pathogen and they will destroy it basically from the inside out. The major histocompatibility complex known as MHC, this plays a huge role. There's 2 different types, MHC1 and MHC2. MHC1 is found in all of your healthy cells that have a nucleus. Remember, your red blood cells do not have a nucleus, so they're not on those. But all of your other cells have a nucleus, you will have the MH1. It is unique to you. It's a protein that is on the surface of the cell. It distinguishes that cell as normal and is what we call self. Major thing with the immune system is being able to distinguish what is you and what is not you. We refer to that as self versus non-self. Excuse me. So the MHC is the marker that's on the surface of your cells. It is unique to you. So mine are different from yours, which is different from your neighbors or family members. They're all different. The MHC2 is only on the surface of certain cells that are known as your antigen-presenting cells. These are like your macrophages, your B cells. This is going to be necessary for the activation of T cells. This is just showing a description or a diagram of what they look like, the MHC2 versus 1. Do you have to know for this class? No. Once again, this is where I go, you know what, this is an intro class. I just want you to know that there is MHC1 and MHC2. And this is involved with determining, MHC1 is involved with determining self versus non-self. because this is going to be critical in how the adaptive immune response works. Your antigen presenting cells, it's often abbreviated as the APC cell. This kind of gets everything started. This will activate your T cells. The T cells, remember, are part of the cell mediated immune division. What happens is these are cells that can carry out phagocytosis, so they're able to recognize, oh, there's something foreign there. How do they know it's a foreign cell? Because it doesn't have the MHC1 that's identical to yours. That's how it's able to cell. It doesn't care whether it's a bacteria. It doesn't care whether it's a fungus. All it cares is it's seeing self versus a non-self. In other words, you versus not you. If it's not you, it will basically eat it. It will destroy it. What it does after it engulfs, it's going to destroy this foreign substance by phagocytosis. Once it has engulfed it, it's going to take the epitope and it's going to use that to start the whole adaptive immune response. So how does it do this? In this case, what you have here are a bacteria. It doesn't matter which kind. So you've got this bacteria, and notice it's got these little red dots on the surface. This phagocyte comes in, and oh, it doesn't see the MHC1 on the surface of the cell, therefore it's not you, so it's going to engulf it. It's non-self, so it's going to engulf it. Fuses with the lysosomes, it's going to start breaking it down. What does it do with those little red circles? Those are the epitopes. Something on the surface it's going to take, and it's going to move it and present it or put it on the surface of its cell. It's like taking a light going to the surface. So this white block, or the antigen presenting cell, is like I'm taking this big old red circle and I'm putting it on the surface here as a flag or something, a signal to say, hey guys, this is something foreign. We've got an invader. Look for this red circle. This is what we're looking for, people. That's what the invader has. Everyone, be on the lookout for this. That's the job of the antigen presenting cell, is to destroy the invader and then signal, let everybody else know you've got an invader, and what is it you need to look for? Find something that's on the surface of the cell, because anything on the surface of the cell obviously is going to be exposed. You'll be able to see it. Don't put something that's internal. Nobody's going to see that. So the antigen presenting cell engulfs the foreign substance. It takes something off the surface. That's what is the epitope. Now, along with this, we have the lymphocytes. We have our B cells and our T cells. They all start in the bone marrow. The B cells will mature in the bone marrow. They stay there. They mature in the bone marrow. They will eventually produce, some of them, not all of them, some of them will produce what's known as plasma cells. And it's the plasma cells that produce the antibodies. The B cells are involved with the humoral immune response division. The T cells will mature in the thymus. Easy way to remember that. B for bone and T for thymus. The T cells are involved with the cell-mediated immune response. There are several different types. There's regulatory T cells, there's cytotoxic T cells, and there's helper T cells. This is a scanning micrograph showing one of your T cells, how it's not smooth. It has those little spikes on it that increases surface area. That's kind of what it looks like. So you had that first step of something foreign came into the body, was able to penetrate, and it's inside the body. It got past the first and second line of defense, so now you've got your third line of defense. The antigen presenting cell has ingested that pathogen and it presented the epitope on the surface of the cell. It put, in this case that we were using, it put that little red circle on the surface saying, hey guys, this is what we're looking for. The antigen presenting cell activates your helper T cells. That's who it's showing it to. Saying, here you go, this is what we're looking for. The activated helper T cell now, number one, it's going to go through cell division, make more copies of it. Everyone's, their sole purpose. This is where the specificity of the adaptive immune response is it a helper T cell that is now activated to look, in this case for this little red circle, is it going to see an orange circle? No, it doesn't care. What about a red square? Nope, doesn't care. It's looking for a red circle. It is very, very specific. And it's going to make copies of itself or clones so that you can start increasing the numbers. We have an invader. We need to go out and search. We need our reconnaissance going out searching and destroying any of these invaders. We don't know how many are there yet, so that's why we have to make a lot of helpers or clones to help us with this. So that activated helper T cell starts going through cell division very rapidly, making lots of clones. All of them are activated and are programmed to look for, in this case, the red circle. Some of these helper T cells are also going to activate your cytotoxic T cells. Now the cytotoxic T cells, some of these will produce memory cells, so that you will never forget that little red circle. What do the cytotoxic T cells do? They're going to go out. You're stimulating your macrophages, your neutrophils, your natural killer cells. Everyone now is on the lookout for that little red circle. Anything that has a little red circle, it's going to be destroyed. You've stimulated memory cells for the helper T cells. As the cytotoxic T cells, the name implies, it is going to go out and be searching throughout the body for this little red circle. The helper T cells are also going to stimulate or activate the B cells. So we've been talking about the helper T cells as part of the cell-mediated immune response. Now they're also responsible for activating the humoral, the B cells. So they do work together. The T cells have to activate the B cells. That just happened. How? By the helper T cells. Which, by the way, just in case as you're reading, there's two different types of helper T cells. There are some that are known as CD4 and some that are known as CD8 cells. That's going to come up later. Your regulatory T cells, they're keeping the helper T cells and the cytotoxic T cells in check. As the name implies, they're kind of helping to regulate all this. So this is just showing the activation of a T cell, that you have your antigen presenting cell here. It's showing the little red marker here. You just activated a helper T cell because it's showing right here. Here's the thing we're looking for, guys. And then what does this cell do? It now activates additional helper T cells. It activates memory T cells. It's going to help activate cytotoxic T cells. And it will also activate the B cells. As the cytotoxic T cells are activated, their purpose is to go out, find the cells. They're going to kill cells that are infected, typically with intracellular pathogens. That's why it's referred to as cell-mediated. It's going to kill the cells by using perforin and granzymes, and they have memory cells. So that if you ever see this again in the future, those secondary responses, it's due to these memory cells. So these compounds that are made, the perforins and the granzymes, the activated cytotoxic T cells, when they come in contact with the pathogen, it releases these. What these compounds do, essentially, they come in and they poke holes in the membrane. And basically all the insides leak out, and that's how it kills the cell. As I said, the helper T cells are also activating your B cells, which is part of the humoral immunity. The B cells, in turn, once they have been activated, they are going to trigger the plasma cells, which is a specialized type of B cell, to produce antibodies against that specific epitope. So in this case, the red ball. It's going to say, okay, we're on the search for the red ball. Make a lot of antibodies specific against the red ball. That's what we are on the lookout for. And also, you're going to make some memory cells. Memory cells are also going to last a long time, so that once again, if you ever come in contact with something that has a little red ball on it again, you can activate right away and have an immediate response. So, excuse me. What happens is, with all of this, you've got all this activation now of the B cells. The B cells, this right here is showing the CD4 is a type of helper T cell. It's activating the B cells, and the B cells are activating the memory cells over here. So you will always remember and know to make these antibodies, and right away, the plasma cells are producing all of these antibodies, which are very specific for the little red dots from our original example. Here's something to think about. So kind of putting everything together. You have a foreign invader. In this case, we're talking about a bacteria. It is engulfed, phagocytized by an antigen-presenting cell, which may be a macrophage. So let's say it's a macrophage. It detects, oh, here's a foreign substance. How did it know it was not part of you? It's because it did not have your MHC1 complex. It looked different. It didn't have that. So it's not you. So it knew, oh, this is something that's not part of you. Let me destroy it. It pulls, in this example we were using, that little red circle from the surface. And it puts that, it presents that on the surface of its cell, which is why it's called an antigen presenting cell. And it's like waving it around. Here, guys, this is what we are looking for. And it activates the helper T cells. The helper T cell is going to make lots of copies. All of them are geared for looking for the little red circle. There will be some memory cells, so that in the future, if you see it, you're ready to go. The current cells, not the memory ones, but the other helper T cells, some of them will then activate the cytotoxic T cells, which are going to then go out, and they know their sole purpose is to look for that little red circle and destroy it. It may activate complement. It's going to activate perforin and granzymes that are going to poke holes in a cell that have it. It's going after it. It sees the little red circle, it will kill that cell. That's its sole purpose. There are memory cells. So once again, if I ever see this in the future, I can respond immediately. The helper T cells also are activating the B cells. The B cells are going to then activate the plasma cells to make the antibodies. The antibodies are going to then go out through the blood in circulation and search. They will bind whenever they see that red circle. And remember, when an antibody binds to it, it can trigger things like neutralization, agglutination, all those steps we previously talked about a few slides back. Basically, it's helping to flag, here it is, I found one, I bound to it, come get it. Somebody come destroy this. Eat it, destroy it. Think about something now. The helper T cells are kind of the main instigator. They activate the T cells, but they also activate the B cells. The reason I'm focusing on this is the helper T cells, as I mentioned just a bit ago, there's two different types. They're classified as either CD4 or CD8. I don't know if you guys have heard of this before or not, especially the CD4 cells. It's a type of helper T cells. The reason I'm stressing this is because the HIV virus, which is the causative agent of AIDS, HIV is the virus, AIDS is the disease. The HIV virus One of the ways that it works is that when it infects the body, it inhibits or disrupts the CD4 cells. In other words, CD4 cells are a type of helper cell. It's turning off the helper cells. It's destroying them. So think about that for a second. You've got a virus that's invading the body. And what it does is it shuts down the helper T cells. And why is that so important? Because it's the helper T cells that activate the other T cells and the B cells to make the antibodies. So if you inhibit, if you prevent and interfere with the helper T cells, you have just shut down your immune system. You've just shut down the adaptive immune system. Because if you knock out the helper T cells, that means you can't activate the cytotoxic T cells to go out and search throughout the body and destroy those cells. You cannot make antibodies because the B cells were never activated because the helper T cells were destroyed. From the standpoint of the HIV virus, that is a very effective way to infect your host and for survival for you. Not good for the host, but from the virus, you just shut down the host immune system, which is the thing that is trying to kill you. From the host standpoint, your surveillance system just got knocked out and you're in trouble. Somebody who has HIV, one of the things they will do is they will monitor the level of the CD4 cells. That is one of the criteria, is what is the concentration of those cells that they use to determine, do you have AIDS yet or not? You can have the HIV virus and not develop AIDS. If you develop AIDS, you are in the latter stages of the infection. Your immune system is being destroyed. Why? Because the virus just knocked out the helper T cells. So like I say, think about that. You see how you knock out one player and it disrupts everything. Like I said, it's a very effective way for the virus to infect us. With the primary response, the first time you are exposed, there is a lag period. There's a delay. You have to get all these players up and running. It takes a while to activate the helper T cell, and it's going to take a while for that one to go activate the cytotoxic T cells and get those out in the blood system. It's going to take a while to get the B cells activated. There's a lag period. Because of the memory cells, when you have a secondary response, it's going to be immediate. You already have that initial work done. So once again, this is showing the primary response down here in the beginning. You have first your IgMs are activated first, but you have a delay here. In this particular example, it's showing a lag period of 10 days. And then after that, you finally get your IgGs. Now, because of the memory cells, look at this. If you have a subsequent exposure, you start to see production in 5 days, half the time. And initially, you see the IgG. Before, it took almost 20 days to peak. So your response is much quicker. And also notice the difference here in the level of response. Oftentimes, the secondary response, you may not know that you even have the infection, as I've said before. This is why sometimes people would say, okay, years ago before the chickenpox vaccine, some people would say, oh, you only get chickenpox once in your lifetime. That's it. Yes and no. The first time you're exposed to it, yeah, you're going to be sick. But because of the memory side of production of the cells, if you are exposed a second or third or fourth time, however many times, any subsequent time, because of the memory cells, they're going to react right away and start fighting. You may not show any signs or symptoms. So, have you been exposed to chickenpox multiple times? Yes, but you only showed the disease symptoms and signs the first time because your immune system was very active and was able to knock it out. With vaccines, oftentimes with vaccines you have to get a booster shot, and some people, especially, this came up a lot during COVID, where some people would say, well, wait a minute, I got the COVID vaccine and then I had to get a booster. Why is it that I felt really, not real sick, but I just felt achy, I had a fever after I got a booster shot? That just means that your immune system is doing what it's supposed to do. You're increasing the number of antibodies that are floating around. Why do you get a fever? Why do you feel achy and stuff? Because your immune response is like, you know what, I've seen this before. Hey guys, come on, it's back, we gotta attack it. You're trying to increase those memory cells, get those numbers up high. It's just the body's response. It's doing, your immune system is doing what it's supposed to do. With this adaptive immunity system, there's different types, the way we classify them. Some people just classify them as natural versus artificial. And then we go active versus passive. So if we combine them together, natural is you are going to be exposed naturally to the pathogen. Artificial, obviously, you're not exposed naturally to it. There's some artificial means to it. Active. Active is when you are exposed and you are going to make the antibodies against whatever the foreign substance is, whatever that antigen is. You make the antibodies, which means you have long-term protection because you're going to have those memory cells. Passive is when somebody else has made the antibodies. You're just kind of reaping the benefits from somebody else. It's providing protection for you right now for the short term, but there's no memory cells. So when you put them all together, natural active immunity. This is when you have been exposed to the pathogen. Let's say you got the flu. Okay, you were exposed to the influenza virus, and I'm so sorry, but you got sick. Now, will you have long-term protection against that particular strain? Yes, you will. Why? Because you produced the antibodies, you produced the memory cells. Natural passive immunity. The classic example of this one is when a mother is producing antibodies and she is breastfeeding an infant and she's passing those antibodies to the child. Does it give protection to the infant? Yes, it does. Does the child have long-term protection? So when, let's say, she's nursing a 6-month-old infant, is the infant getting protection? Yes. Will the child at 5 years of age have protection? No, because there's no memory cells. But mom is providing protection right now for the short term. This can be effective, certainly, on the short term when a newborn does not have a fully functioning, up and running immune system, and so the mom can help provide protection. And some vaccinations can be given right at birth and shortly thereafter, but there's a time period of when you have to be able to give the various vaccines if you do that. Excuse me. This leads into artificial active immunity. This is when you receive a vaccine, when you're immunized against something. You are given, and we'll talk next about vaccines briefly, you will be given a shot that contains basically an epitope. That a piece of the antigen that is going to trigger the immune response. It's going to trigger those helper cells and the B cells, and it will give you long-term protection because you're going to make the memory cells. Do you get the actual disease? No. You were exposed to the antigen, but you don't get the disease, and we are getting much better with production of our vaccines, as we'll see in a moment, to prevent you from getting the actual disease. Artificial passive immunity, this is the least common of all the possibilities. This is where an individual receives pre-made antibodies. So they receive typically an injection or IV. This may be given if someone is immunocompromised, they cannot receive a vaccine. Maybe they're allergic to some of the components. There can be a variety of reasons. They're immunocompromised and there's an outbreak of a disease. You want them to have protection. You give them pre-made antibodies. It will give them short-term protection right now, but not long-term because they're not exposed to the antigens, so they're not making memory cells. There was an instance back in 2014 when there was an outbreak of Ebola in Africa. And there were healthcare providers from all over the world that were volunteering, going there, trying to help treat the various patients and deal with the outbreak. There was an American physician who was working over there, and he contracted Ebola. And he was flown back to the U.S. I mentioned this in one of the previous chapters. He was flown back to the U.S. under very strict quarantine conditions, was taken to Walter Reed Hospital where they quarantined him. They treated him, and thankfully he survived. Mortality rates are very, very high with Ebola. So thankfully he survived. Shortly afterwards, there was a nurse who had been working at the same facility in Africa that also came down with Ebola. So she was also flown back to the U.S. under the same conditions, flown to Walter Reed. She was not recovering quite as well as the physician had. And so then somebody finally realized, wait a minute, he recovered, which means he would have antibodies. So what they did is they took a sample of his blood. They were able to isolate and collect antibodies and increase production of those, and then inject the now sterile antibodies that he made during his exposure and injected those into her. So they were anti-Ebola virus antibodies. And that's what she's suffering from, is the Ebola virus. It worked. She survived with the help from his antibodies. So that's a case of where she was given those pre-made antibodies. It's not nearly as common as the other possibilities, but it does happen sometimes. And so this is just another way of showing these 4 different categories. With vaccines, what you want to do ultimately is you want to reach what we call herd immunity, the maximum number of people to be protected against a particular pathogen so that you can prevent outbreaks of the disease. The more people you get protected, the less chance you have of people getting sick. And that's what you want. There are different types of vaccines. When they first started doing vaccines, which have been around actually for hundreds of years, I don't know if you guys are aware of that or not, but historically they have been. They initially used what we call live attenuated. Attenuated means it's weakened. That's what they used to do. So they would use the entire bacteria or the entire virus. And there's a process you go through, adding chemicals, etc., to weaken it, to attenuate it. Is there a chance that you could develop the disease? Yes. You still get protection from it. Nobody really liked the idea that it's a low chance, but there was a chance you could get sick. So as our technology improved, then we figured out ways of how to inactivate the pathogen, how to kill it or inactivate it so it cannot cause disease, but you can inject it in someone and they can get the protection. And once again, as our technology improved and our knowledge improved, and when we realized it's not the entire organism, say for bacteria, and or viruses. It's just that epitope. It's not the entire outer surface. It's just that epitope that you need that serves as the antigen. So let's do a subunit. Subunit means you don't even have the whole organism. You're just getting the antigen, just the thing that's going to trigger the antibody response. That's what's in the vaccine. If you have a subunit, because you don't have the whole organism, there's no way you can get sick from it. Sometimes you're trying to develop vaccines against toxins. It's called toxoid. It's inactivated toxin. A conjugate vaccine. This is where you have a subunit vaccine with a protein attached to it. Proteins, we have learned, are very effective in triggering the immune response. So remember way back in the beginning we talked about the haptens, where they're too small to really trigger immune response. Well, on a conjugate vaccine, what you do is you take that little tiny piece, but you stick a protein on it, because you know the protein's going to help enhance the response. And that would be a conjugate vaccine. One thing I do want to say about vaccines. Many years ago, there was a report that came out that said some of the chemicals, some of the components in vaccines, that it increases the chance, if a child receives the vaccine, it increases the chance for the child developing autism. And so a lot of people read that and started saying, I don't think I want to get my kids vaccinated. What happened is several scientists started trying, when that report came out, let's see, we haven't ever seen this before, let's see if we can repeat. No one was able to repeat those and get those results. That started raising suspicions on the initial research project. If you did your research correctly, somebody, somewhere, should be able to repeat it. And nobody was able to repeat it. And finally, several years ago, the scientist who wrote the initial article, he came out and he admitted that that entire paper was fraud. He made up the data. Unfortunately, when he published the first paper, it made first page news. When he came out and said, finally admitted that it was all fraudulent, that did not make front page news. Now, he lost his job, and he obviously is no longer respected in the scientific community and will never work again, and certainly will never publish again. So he destroyed his career. Why he did it, I don't know. But there are still some people that misinformation is out there, and some people still see it and they will still believe it. That is not true. Vaccines will not cause autism. That has been debunked for years now. Overall, vaccines, have there been some problems in the past? Yes. We're continually to improve things with vaccines. We have watched a number of diseases, the number of cases of those diseases drop dramatically due to vaccines. Smallpox was the first disease that has been eradicated, completely eradicated worldwide. It does not exist anymore. Why? Due to the effective vaccination program with that. We have been close to eradicating polio. There's 2 different vaccines with polio. We have been trying to eradicate it. The remaining areas of where polio is are in areas in the Mideast, such as in Afghanistan. They're actually in areas where the Taliban and other, we'll just say, political unrest is occurring, and it's not safe to get healthcare workers into those areas to finish vaccinating. So a number of what were considered childhood diseases, chickenpox, mumps, measles, German measles, a lot of those, the number of cases have dropped dramatically. A few of those, like measles, we've started to see a resurgence, an increase in number, and some of that is directly attributed to people who are not vaccinating, like their children. And so in the unvaccinated community, you're seeing an increase in number of cases of people getting some of these diseases. And, you know, it's, we have vaccines that can prevent that. So the specific diseases we talk about in following sections. But this is where you had your innate immunity from the previous chapter, the first and second lines of defense, that the first line hopefully prevents an organism, whether it's a virus, fungus, bacteria, whatever it is, it prevents it from getting beyond our physical boundaries. It can't get through the skin. It doesn't get in at all. If it's able to penetrate, then you have your secondary response. The complement, the inflammation, the maybe the acidity of the stomach. It's so, the pH is so low, most things can't survive. It would kill it there. Or the lysozymes that's bactericidal in your tears. It's preventing it from getting in. But if it does get in, you have your macrophages that are circling around and carrying out phagocytosis. If it manages to get past that secondary line of defense, then you've got your third line, that's the adaptive immune. And that's where it's very specific. This is going to be the production of your T cells and your B cells. B cells activate the antibodies. There are memory cells so that if it ever sees this foreign substance again, it can respond immediately. Everything's about your survival.